The overall goal of this live cell microscopy technique is to study the spatiotemporal events that occur during cytokinesis in the fission yeast model system.
This method can help answer key questions in the cytokinetic field, such as the molecular details of the different stages of cytokinesis over time. The main advantage of this technique is that different spatial components of the cytokinetic machinery can be analyzed over long periods of time with minimal toxicity to the cells.
Though this method can provide insight into cytokinesis in fission yeast, it can also be applied to budding yeast and other fungi.
To begin this procedure, pour 3 to 4 milliliters of melted media laced with vitamin C onto a glass-bottomed culture dish. Once the media has solidified, use a sharp scalpel to gently raise the media slab from the culture dish, and place a pipette tip between the media slab and the culture dish to prevent the slab from falling back into the dish. Next, load 2 to 5 microliters of resuspended cell culture between the media slab and the glass bottom of the culture dish. Then very gently remove the pipette tip and place the media slab back in its original position on the culture dish. Incubate the cells in the culture dish for 30 minutes to an hour, in the dark, at the temperature the microscopy will be performed. When the cells are ready for imaging, place a drop of oil on the outer side of the glass on the culture dish. Place the culture dish on an inverted microscope. Focus on the medial plane of the cells and ensure that they are